Now, there are some people who call themselves transhumanists who say that we can modify our own evolution to create humans that are more than human, that have attributes that no other humans have today.
Yeah. Yeah, they've actually changed their name. I don't know whether this will be a good marketing strategy. They're now Humanity Plus.
Humanity 2.0?
Yeah, so my clear and strong answer to that is maybe and maybe not. We don't understand very much about what makes, genetically, what makes people better. We know a fair amount about genetic variations that give people very low intelligence, pathologically low intelligence. We know basically nothing about genetic variations that give people higher intelligence. So first we got to figure out what the enhancing variations are, if there are any, and then convince people to be willing to use them. We may be able to change some things like eye color and hair color, but intelligence, math ability, musical ability, again, I suspect the best we'll be able to do is a 60% chance of being in the top half.
And we're still talking about changing embryos. We're not talking about changing adults.
No, I mean, although, you know, we do things that enhance born people. The greatest cognitive enhancing tool that humanity's ever come up with is reading and writing. And not only do we allow people to teach their kids reading and writing, we require it. That's a way of changing somebody's cognitive abilities after they're born. You don't change their genes at all, but you change them fundamentally. And I think that leads to a really important point. People get very excited about genetic enhancement and genes enhancing. You know, genes are significant. They're not magic. The difference between the best genome we could probably come up with today and average, I'm willing to bet would be less than the difference between a kid born to a wealthy family in a rich country and a kid born to a poor family in a poor country. And those are completely environmental differences that we could fix now, but we don't.
I understand there's a lot of work being done in stem cell research these days. First, could you just give a very brief description about what is a stem cell compared to other cells?
Sure. There are two interesting things about stem cells. One is they will live forever, or at least for an indefinite period. Most of your cells will divide 40, 50, 60, 70 times and then die.
You mean the stem cell lives as long as the individual lives?
Yes. So we know that they can live at least 123 years because the oldest known human lived to be 122 and a half. And so you add on the 9 months for pregnancy, 123 years. So they don't die off after a limited number of cell divisions. But the other thing is they will turn into different cell types. So an embryonic stem cell, you and I started out as a small number of cells in the middle of a 5-day-old embryo, poetically called the inner cell mass. Those cells, which make up human embryonic stem cells, can make every cell type in the body. We know that because they did, for you, for me, for all of your viewers. What makes it exciting is you could then take those cells and make them into heart muscle cells, brain cells, kidney cells, in a way that adult bodies don't do. So that's the important thing about stem cells.
How do you tell it to do that? Every stem cell has the capability to become any specialized cell, but how do you make it become the one you want?
As always, it's a little more complicated. There are stem cells that are halfway down, so there's blood-forming stem cells. They make all 100 different types of blood cells, but they won't make kidney cells or brain cells. So if you start with a blood-forming stem cell, you're going to get a blood type, a type of blood cell. Which type do you get? That's what the research for the last 15 years has been focusing on, with some success, but not perfect. Basically, you put them in environments where they're getting chemical signals from other cells that tell them this is what you should become.
Now, in a baby that's gestating in the womb, for example, every cell seems to know its place. You have the right amount of muscle cells and the right amount of bone cells, and they're all in the right place.
Well, if you get far enough to actually be born alive, most pregnancies, most fertilized cells never become babies. Most fertilized eggs never become babies. Maybe because something goes wrong, and maybe it's something that goes wrong with that system. So early on, an embryo divides into, the stem cells divide into 3 different cell types that make different kinds of tissue. And then later on, if you're in the part that makes a brain cell, where it is in the brain and what signals it's getting from the things around it help determine what kind of brain cell it turns out to be. But we're still trying to figure that out. That's where the frontier of stem cell research now is trying to make these cells become what we want them to be. We can do it for some. We can do it occasionally for others. We're having trouble doing it at all for other types.
Let me ask you this question, which is a slightly different topic. So I've heard that it might be possible to revive long extinct species. Like maybe you could find a dinosaur bone and find a few strands of DNA in that and somehow manage to merge that into a live cell of some creature and grow new dinosaurs.
Maybe you could revive liberal Republicans.
Well, I wouldn't go that far.
Dinosaurs are out. I don't know about liberal Republicans.
Okay.
We're not going to be able to do dinosaurs. The last dinosaur, unless you count birds, which are kind of surviving dinosaurs, died off 66.4 million years ago. No DNA is going to be left. The Jurassic Park trick of the amber was clever. People have looked, doesn't work. But Pleistocene Park, that's really quite possible. We have done whole genome sequences of animals that died 200, 300,000 years ago during the Ice Age. The DNA is preserved, so we know their genomes. We know the genome of the woolly mammoth. And people are talking about reviving, maybe not exactly mammoths, but something like mammoths by taking cells from an Asian elephant. Turns out mammoths were closer to Asian elephants than African elephants. And using CRISPR-Cas9, which we talked about earlier, editing those cells, sequence the Asian elephant, sequence the mammoth, see where they're different, edit the Asian elephant cells to make them like mammoth cells, and then turn that into a baby and hope that an Asian elephant mother will give birth in a very confused way probably to a really hairy little elephant calf that's a mammoth.
Do you think like this process would need to be regulated somehow, or people who could afford it should just go ahead and
I think there should be some regulation. I don't think we should be terrified of it, but you wouldn't want to be the person who unleashed a new invasive species on the planet. Kudzu is a real problem in the southeast. Clams, the Chinese clam is mussel is a problem in various places. You don't want to do that, so you need some regulation before there's environmental release. And you also don't want to kill off too many mother elephants. You want to make sure this is safe for some of the animals involved.
We just have a few seconds left, so let me ask you this last question. Are you optimistic that all this research is going to lead to something good for humanity in general, or do you think maybe we're treading on dangerous ground? And you have about 5 seconds to answer.
I'm optimistic. We will screw up occasionally, but on balance, this will be a good thing.
Okay, very good. And on that note, I'd like to thank you for being here today.
My pleasure.
Hank Greely, Director of Law and the Biosciences at Stanford University. I'm Marty Wasserman for Future Talk. Visit our website, www.futuretalk.net. For Future Talk, we'll see you next time.